In question number 6, you are asked to find efficiency of Meissen engine, assuming that delta G of reaction, ATP hydrolysis under physiological conditions is negative 45 kilojoules per mole. Here we compare Meissen with engines, and for any engine, you can calculate the efficiency by dividing the useful work that this engine can make to the total energy that it spends to do this work. And here's the formula. This is your work, this is your total energy available. In our case, the total energy available can be calculated from the ATP hydrolysis reaction. And now let's take a look at the work that Meissen head does when it translocates along the thin filaments. So from physics, you should remember that work equals force by the distance that this force is applied to any object. So we know the force. Force is given to us in piconewtons, right? This number. And the distance isn't given, but you can find it in your textbooks. So it's either 10 or 11 nanometers, depending on the source that you use. In ebook, you see 11 nanometers is the distance of translocation of Meissen head along thin filament per 1 power stroke. So we pick up this number, we multiply 4 newtons, piconewtons by 11 nanometers, and we get joules. So this is per 1 molecule. Okay. Now the ATP hydrolysis energy is given per mole, negative 45 kilojoules per mole. So I'm converting kilojoules in joules, divided by Avogadro number, and get the available energy per 1 ATP hydrolyzed. Okay, and now what I need to do is just to divide these two numbers. Okay, and I convert this number to positive because efficiency cannot be negative. So meaning that this is energy for ATP hydrolysis, available energy. It's a positive number now. And then the Meissen efficiency equals about 58.9%. This is the answer. Question number 7 in homework 2 asks you to find the Hill constant and the P50 for a blood sample. So this is a practically significant task when you measure the dependence of fractional saturation of your given blood sample or given hemoglobin sample from oxygen pressure. And these are experimental data are given in this table. So now, in order to solve this problem, you have to use the Hill plot equation, in which the logarithm of this expression, where y is fractional saturation, equals n by logarithm PO2 minus n by logarithm P50, right? So you have to memorize this equation in this course, and as you can see, it's a linear function where your y is this logarithm of this expression. This is your y, and your x is a variable logarithm of PO2, right? So if you know the dependence, then you can find your n as a slope of the line, and you can find, in general, you can find P50 as an intercept point, right? So now, in order to plot this data points, you have to calculate your x and y. So your x is calculated based on the data of oxygen pressure, right? So you just take logarithm by base equal 10, and your y is a logarithm of fractional saturation by 1 minus fractional saturation. So this basically you use these numbers to find the corresponding numbers, right? So now you know your x and y, and what you do, you have your x and have your y, so which are also shown here as logarithmic functions. You plot the data and you draw a line, best fit line. And now from this best fit line, you determine the parameters. So what I've done, I measured the slope here. I measured this distance and divided it by, I called A, and divided by this distance, I call it B. Okay, this is my slope of the line, and this slope corresponds to n. So this is my slope, right? So that's the way I found Hill coefficient. It's about 2.8, right? Now the P50 value in general can be found as an intercept point with y-axis, but it's far away, it's not convenient to determine this. That's why there is another strategy here, easier strategy. It's a sort of intercept point, simply the intercept point of this line with x-axis right here. So this is 74.1 torr. And let me explain why. So I already explained it in the lecture, but just to reiterate the point. The intercept point on this graph with x-axis will correspond, obviously, y equals 0. So your logarithm of y02 minus divided by 1 minus y02, this value is 0. So this turns into 0. And once it turns into 0, you get n cancelled, and you transfer P50 on the other side of the equation. You unlock the equation, and you get the simple relationships PO2 equals P50. So basically at this point, your PO2 equals P50. This is P50. So this is a trick that is very useful in this case. And these are your answers here. N, Hill coefficient, and P50, 74.1 torr. Thank you. Question number 8 asks you to draw the complete chemical structure of fragments boxed in 1b and indicate the same fragment in 1c. What you have under A is the primary structure of ribonuclease A. Under B, it's a secondary structure, and under C, it's a tertiary structure of the same enzyme. So RNase A is the same RNase that catalytic mechanism of which we study in this course. In order to draw a complete chemical structure of this fragment, you first have to identify which amino acids here and here are involved in hydrogen bonds formation. And I don't want you to look at these labels which are not clear on the slide. Instead, you should identify the N-terminal amino acid, which is here, K, and then go through the chain and find out which amino acids are part of the structure by just simply counting them in the primary structure right here. So once you identify which amino acids are here and where is the end, so this is N-terminal end and this is C-terminal end, you can go ahead and draw your peptides as they are. So I have my wiggly line right here indicating that this is just a fragment of the peptide and it continues with other structures and somehow it is bound to this end and it's bound to this end, right? So basically this is my beta sheet structure, and then I indicate the hydrogen bonds between hydrogen bond donating groups and hydrogen bond accepting groups. So please keep in mind that these arrows that are shown here, they indicate only hydrogen bonds found in the backbone. No side chains are involved in these hydrogen bond formations. So that's the definition of secondary structure. You can see that sometimes there is a deviation from a normal anti-parallel beta sheet structure as we learned it. For example, this chain, these amino acids, they form a hydrogen bond within a single chain, and that's what is reflected actually here. So this NH forms a hydrogen bond with this CO group, right? Now, in order to find out where this fragment is located on the structure, I would go after valine residue and indicate that this valine, this is a C-terminal end with a valine on the as a last amino acid. How do I know that? Look at this structure. This is a beta sheet structure, right? And if you take a look at the other end, this end, it forms alpha helical structure, not beta sheet. So these are different ends in terms of secondary structure. And here on this picture, first of all beta sheet structures, they are indicated in cyan color, right? And alpha helical structure is indicated in red color. So this is definitely a beta sheet structure, right? And meaning that this is C-terminal end. So my valine is this, and it goes this way. And now all this is a beta sheet that is not boxed. And now after that, we have a beta sheet, a three-stranded beta sheet that is boxed, right? So the three-stranded anti-parallel beta sheet is boxed on fragment B, and this is actually the only three-stranded anti-parallel beta sheet structure found in the entire structure of ribonuclease A. So I circle this in my answer. Question number 9 provides you experimental data for a catalytic reaction. And of course, we follow Michaelis-Menten catalysis, in which enzyme binds substrate to form enzyme-substrate complex and then produce product. So, in reaction, there are three reactions listed here, and this velocity of the three reactions will measure it under different substrate concentrations, which are shown here in micromoles, and these numbers were found in micromole per minute, meaning that this concentration of products was accumulated in 1 minute for each reaction. Here on the slide you see the equation for double reciprocal plot in which inverse initial velocity depends on the inverse concentration of the substrate. So our inverse substrate concentration is x in the linear equation, and our inverse initial velocity is an y in this equation, right? So now we can find the parameters of Km and Vmax. That's our goal, to find the parameters, because that is what is important. And the way we find the parameters is inverse Vmax is an intercept point with y-axis, and inverse and negative Km is our intercept with an x-axis, right? Okay, now the first step to plot the data is to convert this table into inverse values. In other words, find your x and 3 y's. So you basically inverse each number and fill this table for the x, and you do the same for the three reactions in the absence of inhibitor, in the presence of inhibitor A, and in the presence of inhibitor B. So there are 3 reactions. You will eventually end up with 3 lines. And then there is a bunch of questions to answer about this plot. Okay, so let me show you the answers. So this is the plot. So whenever you draw a plot, don't forget to label the axis. This is actually the first important step to do. So this is inversed initial velocity, and this is inverse substrate concentration. 3 lines: no inhibitor line, inhibitor A line, and inhibitor B line. So the first question that asks you on the B is to determine Vmax and Km in the absence of inhibitor. So let's take a look at the line in the absence of inhibitor. You take this number, make sure that you pick up the correct number on in the scale, right? It's an inverse scale, and you simply inverse it. 1 over this number is going to give you Vmax, which is here. Don't forget to add units. This is important. Sometimes units could be different, so you have to match the units found in the graph with the units given you in the question. So, and then second intercept point for no inhibitor is here with y-axis. You pick up this point, make sure that you follow the scale and make it negative inverse, and you get this number. Okay, you do the same for the lines in the presence of inhibitor A and in the presence of inhibitor B, right? Then determine the types of inhibition. I think here you need to just simply memorize. There are 3 cases of different mechanisms of inhibition. So for competitive inhibition, you have lines intercept on the y-axis. So, but you don't have here these lines. No lines intercept on the y-axis. No competitive inhibition. Then for uncompetitive inhibition, you have parallel lines. So you do have parallel lines. Inhibitor B is parallel to no inhibitor. So your inhibitor B appeared to be uncompetitive inhibitor. And then mixed type of inhibition, they intercept somewhere on this left side of your graph, and you have such a case here. Inhibitor A is your mixed or non-competitive type of inhibitor. And the last question in this problem is to find dissociation constants of enzyme inhibitor complexes. 2 constants are asked for each inhibitor, but in fact there are 2 of them only for non-competitive or mixed type of inhibition, and for uncompetitive inhibitor we have only 1 constant. Okay, let's take a look how we determine Ki prime in uncompetitive inhibitor case. So just to remind you the slides from the lecture. So when we deal with uncompetitive inhibition, our enzyme substrate complex forms a complex with inhibitor, and this tertiary complex doesn't produce any products. Okay, and then when we insert these 2 parameters, Ki prime and Ki prime and inhibitor concentration into the equation, Michaelis-Menten equation, we are going to get an equation in which both Km and Vmax are modified by alpha prime. Okay, and alpha prime is a parameter that combines actually all the characteristics of inhibitor, both inhibitor concentration and Ki prime. So the inhibitor concentration in our case is given in the problem. Let's take a look. So the problem states that both inhibitors A and B are present at the concentration 0.1 millimolar. So what it means? It means that Ki can be found from this equation if you know alpha prime. Now the question, how to find alpha prime, this parameter. And alpha prime, in case of uncompetitive inhibition, is found as a ratio of Vmax and Vmax apparent. So this is the easiest and the most fast way to find alpha prime. So basically, you'll take a look at the intercept points for uninhibited and inhibited reaction. Let's go back to our lines. So we have a line for non-inhibited reaction right here. So I will actually determine already Vmax for non-inhibited reaction, 6.67 micromolar per minute. And we take a look for our inhibitor B. For inhibitor B, Vmax is 4 micromolar. So we find this ratio, and we determine, by this ratio, we determine alpha prime. Okay, we then plug alpha prime in this equation and find Ki prime from this equation. Okay, and here this number was found for uncompetitive inhibition. Now, how do we do with the most complex case of non-competitive mixed type of inhibition, where two inhibition constants have to be found? So we do pretty much similar, we start pretty much in a similar way. So what you see on the slide, it's a cartoonish representation of mixed type of inhibition, where our lines cross at the left portion of the graph. And we start with the same. We find alpha prime. Don't look for alpha first. Find alpha prime, same way as we just discussed before. So once you find alpha prime, you can go ahead and immediately find the Ki prime, same way as you've done for an uncompetitive type of inhibition. Now, how do we do with what do we do with alpha? So alpha is a parameter that contains the Ki, the other inhibition constants that we need to find, right? So we do have the concentration of the inhibitor, and the only thing that we need to know to find Ki is to find alpha. So, and the alpha should be found from this expression right here. So we already know alpha prime. We calculated it earlier, okay? We know Km and Km apparent. So let's go back to our graph. We know Km, Km is this one, and Km apparent for the inhibitor A, right? So these numbers should be plugged into this equation, Km, Km apparent. Alpha prime we know, and we find alpha. Once we find alpha, we find Ki. So this is the answer for inhibitor I. So we provide two answers, Ki prime and Ki. And the last question, number 10, asks you to draw the catalytic mechanism of acetylcholine esterase. So the substrate of acetylcholine esterase is shown here. So first of all, I would like to explain you the physiological significance of acetylcholine esterase. So what you see here is a neurotransmitter released in a synaptic cleft between presynaptic membrane and postsynaptic membrane during signal transduction. So what happens here, the neurotransmitter binds to the postsynaptic membrane and triggers action potential. In fact, the rate of signal transduction, in other words, how many signals can be transduced during 1 second, let's say, depends on how quickly the neurotransmitter is cleared out from this synaptic cleft. And this synaptic cleft contains enzymes that degrade neurotransmitter. And in case of acetylcholine as a neurotransmitter, you have acetylcholine esterase that acts to remove neurotransmitter from the synaptic cleft. So the quicker the enzyme acts, the more signals can be transduced in a second. So basically what I've done, I already answered this question, explained why the high efficiency of enzyme is physiologically crucial. So now, how do we draw the catalytic mechanism of acetylcholine esterase? We know that like serine proteases, this enzyme contains aspartic acid histidine serine catalytic triad. What we do, we just draw this catalytic triad, serine, histidine, aspartic acid, and place next to it acetylcholine esterase. If you take a look at this group, this is ester group. It is very similar to amide bond, amide linkage, peptide linkage found in proteins, and it will be hydrolyzed based on very similar mechanism. So first we have nucleophilic attack of serine residue on CO bond, on carbon of CO bond, followed by formation of tetrahedral intermediate. Then we have acyl intermediate and choline residue released from the active site, and so on. So we go through the stages that we know for mechanism of serine proteases. Okay. And then the last question is to propose an efficient inhibitor of acetylcholine esterase. So now I have pyridostigmine as an answer. However, so this pyridostigmine was taken from internet because I know that this compound is an efficient inhibitor of acetylcholine esterase. And you can find the similarities between the structures of the two. So both of them have positive charge on the right portion of the molecule, right? And then we have some similar structures in the center of the molecule, this the part that is cleaved. However, the structures are not identical. They are different in size, and perhaps due to the different location of carbonyl CO bond in the active site of acetylcholine, we don't have an efficient cleavage of this substrate analog. So that's why it is an efficient inhibitor that is bound by the enzyme but is not cleaved. So now, in order for you to answer this question, you have to draw you could draw an analog of either acetylcholine, like this one, or the transition state analog, which is a tetrahedral intermediate that presumably will bind tightly to the active site. So basically, you don't have to give, you don't need to provide a structure that is found online. You have to come out with your own structure. What is important, I guess, in the structure is that you know that the compound is stable. So all bonds are stable under physiological conditions.